Today we are going to learn about DNA polymerase chain reaction. Polymerase chain reaction is one of the most frequently used genetic technique. PCR is a way of amplifying a small, generally less than 10 kb region of DNA, the target DNA sequence, allowing us to make an essentially unlimited number of copies of that DNA without cloning the region. It can polymerize nucleotide sequence using polymerase enzyme. It is chain reaction because reaction procedure has sequential events which keeps going on and on. Once generated, these copies could be cloned, separated using gel electrophoresis. PCR is modification of DNA replication. Kary Mullis received part of the 1993 Nobel Prize in Chemistry for his invention of the polymerase chain reaction. Before knowing about the process, this is the typical PCR mix generally used by labs. PCR is carried out using a PCR machine or thermal cycler which takes samples through a series of carefully controlled temperature changes for very specific periods of time. First of all, to amplify a specific target DNA sequence using the polymerase chain reaction, starting with template, which is generally double-stranded. And this template can be large and complex. It can even be entire genome. Two primers, which are forward and reverse, are designed and synthesized to make the desired polymerase chain reaction possible. These primers must be complementary to the two ends of the target DNA sequence to be amplified. The primers are added to the template DNA along dNTP precursors and a buffer, and reaction mixture is heated to 95 degrees centigrade for 1 minute. The heat denatures the DNA to single strands. The reaction mixture is allowed to cool to a temperature of 54 degrees centigrade for 45 seconds, at which the primers will anneal to the template. A heat-stable DNA polymerase is added. Such enzymes have been isolated from bacteria or archaea that have evolved to survive in very hot environment. Here, the popular one is Taq polymerase, isolated from Thermus aquaticus. DNA polymerase, that is Taq, extends each of the primer from their 3' end at 72 degrees centigrade. After a specified amount of time for the DNA synthesis step, the denaturation step is repeated at 95 degrees centigrade and the mixture is cooled to allow the primers to anneal. PCR optimization is needed to avoid amplifying background products. The factors we are going to discuss are buffers, magnesium chloride, primer design, cycling conditions, and cycle number. Most buffers have only potassium chloride and trace. Potassium chloride facilitates primer binding, but concentrations higher than 50 millimole inhibit Taq polymerase. Some detergents like gelatin, glycerol, Twin 20 all are used to enhance the specificity. Magnesium chloride is required for primer binding. It affects melting point of template DNA, product and primer template associations, product specificity, enzyme activity and fidelity. dNTPs primers and template chelate and sequestered the magnesium ion, therefore concentration should be higher than dNTPs because these are most concentrated. Excess magnesium gives non-specific binding, and on the other hand, too little magnesium gives reduced yield. So in case of PCR optimization, these factors are taken care of. It is specific to sequence of interests. Length must be within 18 to 30 nucleotide. Annealing temperature is from 50 degree to 70 degree, but ideally it is taken as 50 degree centigrade to 63 degree centigrade, and the GC content should be 40 to 60%. 3' end is very critical. because GC clamp or the G or C, that is guanine or cytosine at 3' terminus, ensures proper binding. Inner self complementary must be less, otherwise it will form the hairpin structure. And the 3' complementarity is very much important. Denaturation and annealing temperatures. These two factors are very important in case of cycling conditions. In case of denaturation, some Taq polymerases require initial denaturations. This is called as hot start. And in case of annealing temperature, less than 5 degrees centigrade, the melting point of melting temperature of primers. If annealing temperature and the melting temperature of the primers are equal, then the binding is not possible. Decrease in annealing temperature results in non-specific binding, but increasing annealing temperature results in reduced yield. And the last one is cycle number. Typically, the PCR amplification cycle is repeated for 30 to 35 rounds, and the theoretical yield is 2 to the power 9. But practically, the output is different because half-life of Taq polymerase is 30 minutes at 95 degrees centigrade. Therefore, if you use more than 30 or 35 cycles at denaturation time of 1 minute, the Taq will not very efficient at this point. The term plateau effect is used to describe the attenuation of the normally exponential rate of the product accumulation in PCR. The application of PCR has been found in molecular identifications, as in molecular archaeology, molecular epidemiology, molecular ecology, DNA fingerprinting, classification of organisms, genotyping, prenatal diagnosis, mutation screening. On other hand, sequencing, as in bioinformatics, genomic cloning, human genome project, and also in case of genetic engineering, as in site-directed mutagenesis and gene expression studies. There are several molecular techniques as RFLP, REP-PCR, RAPT, CFLP, AFLP, and many more. Nowadays, PCR becomes necessary for all kind of molecular genetic works. So that's it for today's discussion. If you have any query regarding this class, please comment. Any kind of suggestions are also welcomed. Thank you for your patience. Please do not forget to like and subscribe.